All right. So it's my very distinct pleasure to introduce the moderator of today's webinar, Antoine de Moray, who is currently an assistant professor at the Department of Biomedicine at Aarhus University in Denmark, and currently in the Bay Area. His lab investigates the mechanisms that enable adult stem cells to regenerate tissues, and results from his work has been published in high-profile journals, including Nature and Science, which resulted in him receiving several international awards for his research and leadership, including an MDA development grant. In addition to his scientific career, he's also regularly teaching communication and resilience to science trainees, and co-wrote the book Magnetic: How Great Leaders Persuade and Inspire. Antoine holds a PhD from Leiden University in the Netherlands, a postdoctoral certificate from Stanford University, and an Ignite certificate from Stanford University Graduate School of Business. And with that, Antoine, thank you so very much for being here today, and the floor is all yours.
Thank you, Renata, for that kind introduction. It's my pleasure to in turn introduce our speaker today, Dr. Sonja Billerbæk. Fluorescence measurements are key to many assays in biology. However, while different assays can report on one single probe, the fluorescence measurements are often depicted as arbitrary units, and this complicates comparison across assays or even experiments. Today, we'll hear about methods to calibrate fluorescence measurements to enable such comparisons. With us is Dr. Sonja Billerbæk, Assistant Professor at Groningen Biomolecular Science and Biotechnology Institute at the University of Groningen in the Netherlands. Sonja trained in the field of synthetic biology and worked on the concept of minimal cells. And during her PhD at the ETH of Zurich, she developed gene replacement methods to enable directed evolution of synthetic cellular systems. With this knowledge in the backpack, she moved to Columbia University in New York for postdoctoral training, and here she developed ingenious biosensors using yeast. For example, she modified yeast so that it expresses REC pigment in response to receptor activation, thereby effectively enabling the replacement of expensive antibodies with cheap yeast cells for the detection of, for example, pathogens. Now in Groningen, her lab seeks to further leverage the biology of yeast by investigating its ability to produce antifungal drugs. Her work has received competitive awards, including a Simons Foundation Junior Fellowship, and has been featured in Science and The Atlantic. Sonja is a co-founder of SynBioNL and an associate editor of Oxford University Press's Synthetic Biology. Sonja, thank you for joining us today. We look forward to your talk, and the floor is yours.
Thank you so much, Antoine, for this very kind and elaborate introduction. And it's really a pleasure for me to be here and kind of spread the word on the calibration work that we have been doing over the last couple of years. It's not really the focus of my laboratory. As you just heard, I work on yeast and antifungal proteins at the moment, but you will see how this relates and who has done this work. So let me just move all these things here. So the webinar in a nutshell, we will talk a lot about typical experiments that we do in synthetic biology, which is E. coli cells expressing a fluorescent protein, for example, a green fluorescent protein, measured in a 96 well plate. And what I will talk you through, how you can effectively come from reporting these measurements in arbitrary units, as for example, fluorescence over optical density at 600 nanometers, into calibrated units measured, for example, as equivalents of molecules, equivalent, sorry, I'm just trying to make my screen a bit more visible, equivalent of a calibrant over cell count, and how you can do that in an easy, cheap, and high throughput manner. And I will hopefully convince you that this can lead to more lab-to-lab reproducibility of these data, to the ability to reuse data directly by other users, by other researchers, and that it can give you additional scientific information. And what I will specifically talk about is really optical density to cell count, and we will talk about calibrants for green, red, and blue fluorescent proteins. So that's kind of the scope that I will talk about. And I will also kind of put that in context, why this is even important for synthetic biology. I will, of course, talk about how our protocols look like and how we calibrate, and also how we actually verify that the calibration protocols we developed are that those are any good. So how kind of we compared them across the globe. And yeah, this already brings me to the next topic. So who did the work? So this work was not specifically done in my laboratory, but it was really a team and community effort that is placed in the context of IGEM. So IGEM stands for the Internationally Genetically Engineered Machine Competition, and it is the premier student competition in the field of synthetic biology. So a lot of universities around the globe have actually teams of about 10 to 15 undergraduate or postgraduate students, and they are supposed to build a synthetic biology project over the summer within a couple of months, and then they compete with their projects against each other during an international competition. And there's a lot of scientists and jurors and so on involved with IGEM, including the so-called engineering committee. So the engineering committee is a committee that helps these IGEM students to actually perform their work. And the initiators of this whole calibration work were really Jake Beal, the chair of the IGEM engineering committee, and Tracy Haddock, who was at the time the director of IGEM of the IGEM competition. And they thought that this IGEM competition would really be a great platform to develop these kind of more calibrated measurements. Once, because we have access to a lot of labs around the globe, and two, because the students that are participating in IGEM are, of course, the next generation of synthetic biologists. So it would be a great opportunity to already train them in thinking along and kind of calibrating their measurement units. And the work was initiated by these two, Jake and Tracy, but then also the IGEM engineering committee members, which is just a group of volunteers, PhD students, postdocs, PIs. I'm also one of them. That kind of help to coordinate all these efforts and do the science. And then, of course, over 300 undergraduate student teams from around the globe that over the years participated in these so-called interlab studies that I will talk about a bit in a minute, that helped us to kind of validate our protocols. So why are calibrated units important for synthetic biology? And here it's good to think about the so-called design, build, test, learn cycle that we use to kind of engineer our biological systems. So for those that are not familiar with synthetic biology, in brief, it's a discipline that tries to redesign or rebuild biological systems, either from scratch or using existing systems. And as biology is relatively difficult to straightforward engineer, so it's not like building a computer where you kind of put together known pieces and eventually they work as expected. Biology has a lot of black box behavior in it. So what we usually do, we design a system as good as we can using prior knowledge and computational modeling, but often we design many different versions of a possible system. We build these versions, we test them, we fish out the one that kind of work as we want, we learn from them, and then we redesign them. And we try to go through that cycle many times, such that eventually we arrive at our final system. To give an example about this, a typical example is a genetic circuit, like this toggle switch, which is built from basically two repressors, CI and LUDOX, and they basically repress each other's expression. And they're also controlled by external triggers, for example, the small molecule IPTG or heat. And depending on how you set the stage and which kind of inducer you add, you can toggle between expressing a fluorescent protein. So for example, the promoter that drives one of these repressors also drives the expression of a fluorescent protein. And then dependent on the state the cell is in, you can kind of toggle between expressing fluorescent protein or not. And building the circuit has actually a lot of different parameters that influence the dynamic of the switch and even influence if it will work at all. And this is, for example, the strengths of the promoters that drive the repressor expression, the IPTG concentration, temperature, half-life of the GFP, and even the strain, the specific E. coli strain you use to implement that, and a lot of more parameters. So this is just an example to give that usually we include all these parameters. We build a lot of different systems combining all these different parameters. We build them and test them and so on. And the discipline of synthetic biology over the last 10 years has a lot focused not only on building new biological systems, but also building resources for each of this step that enable us to go through that cycle the least times. So for example, we have a lot of computational tools, we have part libraries, we have effective methods to kind of like build our designs in an effective way. And then we have high throughput ways to test them, and also computational ways to learn from them and kind of iterate. And where this calibration method comes in context is really in this test phase here, where we want to work with calibrated units such that these tested devices can actually be effectively compared across laboratories and effectively fed into the learn cycle. And ideally, it means that not every lab has to actually test every device, but rather that you can use data that other people have created and feed them into your learn. So this is really kind of the context where these calibration methods that we have been developing over the time fit in. So what I will talk about in this webinar is really the time course. So you see that arrow here, the time course of the development and verification of the so-called IGEM calibration protocol. So there is likely other calibration protocols out there, but the IGEM calibration protocol is the one that we have been building within the IGEM community, and we think it's reasonably good, and that we're confident that it should be adopted by many researchers. So this is why it's also great to talk about this in this webinar, because it would be great if we would see more research actually reporting not arbitrary units, but rather calibrated units. So specifically, I will start with a study that was published in 2018, where we calibrated green fluorescence and actually used this also as an opportunity to really ask the simple question if unit calibration actually matters for measurement precision and how precise we can measure. And this was verified with the 2016 IGEM Interlab. After that, I will talk about how we can actually precisely calibrate cell count, comparing different ways to do that. And this protocol was verified or validated with the 2018 IGEM Interlab. And then after that, I will go further, not only calibrating green fluorescence, but rather blue and red fluorescence. And this was a study published in 2022 using a 2021 mini interlab. So this was a mini interlab just featuring different labs within the IGEM engineering committee, because during the pandemic, it was very difficult to send stuff and organize a real interlab. And then after that, I will just make you aware, or wanted you to make you aware of an additional publication published in 2022 that is not part of the IGEM protocol, but which we believe is really a great way to go even beyond what we have been developing in this IGEM protocol. So just shortly, the IGEM Interlab studies, what is this? So as I said before, it allows us for protocol validation because we use the IGEM competition. It's a yearly competition to really get access to a lot of teams that are interested in student teams that are interested in synthetic biology around the globe. So here's a map of the 2016 Interlab study. There were 92 teams participating. So this involved 20 different countries. And these teams were sent materials so that they could actually work with. They were sent protocols so they would know what to do. And they were sent data submission sheets so that we could actually collect the data from them. And then also instructions, of course, how to put everything together. And why this is great for us, it is really a tool to, as I said, get access to many labs, many different instruments, different expertise levels. So also, as I said, there is the bachelor students, sometimes master students, also different interpretations. So often cultural differences or language skills, or even, the cultural background decides how you interpret a protocol. So it's really a great way for us to validate all the protocols that we had been developing, let's say, within our kind of rather PI-ish minds. With no further ado, I will start with the first study. Does unit calibration matter for precision? And the reason we actually wanted to ask this relatively simple question is because it could be that the arbitrary units that fluorescent measurements are usually reported in could actually be just not problematic. So it could be that researchers tend to use similar arbitrary units and that actually calibration is not really necessary. It could also be on the other hand that maybe obtaining and using independent calibrants is too difficult, unreliable, or maybe too expensive relative to the value of reproducible units. And again, asking these questions is important because in the end, standardization is all about communication. So it really matters that we all as a community agree on standards. So there's, often not, the consensus is often made, through human interactions. So if you want to convince a community to kind of really do an extra effort to standardize a unit, they should really know that this is, practical, useful, and really matters to get more precise results. So that was the reason we asked these simple questions. So in the 2016 IGEM Interlab has had 92 teams, and these teams were sent a measurement kit, protocols, and data submission sheets. And part of the measurement kit was plasmids. So these teams were given 5 different plasmids, expressing GFP under the control of a strong, medium, and weak promoter. And they were also given a positive and a negative control. The positive control was a known very high expression level promoter that would yield very high expression. And the negative control was just a promoter without a fluorescent protein, meaning the cells would be non-fluorescent. The teams were also given calibrants, and this was really shipped to the teams by mail. And the calibrants were fluorescein isocyanate, so FITS, let's say a fluorescent dye that is available from Sigma, to calibrate green fluorescence. So people had done that before, so we thought that's a good idea. And the teams were instructed to basically make a serial dilution with this and submit the data so that we could compare their fluorescent measurements to FITS. And then for optical density, they were given LUDOX HS30, which is also a compound that is available through Sigma. It is basically colloidal Sigma in water, 30% solution of colloidal silica in Sigma, and it allows to calibrate for instrument differences in the determination of optical density. So it doesn't allow to actually count cells, but it allows to account for the differences in spectrophotometers. And I will actually come back to that in a bit, what that actually means. And then they had to perform a specific experiment, very simple. So they had to transform the plasmids into cells, and then they had to pick 2 colonies each, do an overnight culture. And then the actual experiments were where they grew these 2 colonies in liquid culture, and they took samples before, or like right after dilution, so at time point 0, and then after 6 hours of growth. And they were advised to pipette 100 microliters into a specific well. So they had given we had given them kind of a pipetting scheme. And then they were advised to measure that and submit it for data processing. And we processed the data in 3 different ways. First, we used just arbitrary units, meaning the fluorescent values they had given us, which is often a random number that is generated, not a random number like a measurement, but the numbering depends on the gain and the instrument specificities per optical density at 600 nanometers. Then we also measured normalized units. Given we had a very bright positive control, we could basically report the arbitrary unit as % of reference, meaning we kind of just divided each sample by the fluorescence of the positive control from the same replicate. And we used comparable units, meaning micromolar of the FITS per OD, meaning the instrument calibrated OD that could be generated using the LUDOX. And what happened here is, or what we then did is we compared the measurement precision of these 3 data processing types using the geometric standard deviation across the data. And what you can see is that for arbitrary units, but interesting also for the calibrated unit, we got very large geometric standard deviations of 20-fold, for example. And 20-fold really means that the confidence interval of these data really spreads over 5 orders of magnitude in size, meaning there is really no way to say what the real measurement is. So the standard deviation is just like very wide. If we normalized it, so we kind of like did the percent to the positive control, this geometric standard deviation became much better. So, but something we learned actually from this calibrated units is that most of the data sets were actually close to each other, and some of the data sets seem to have obvious process failures. So process failures might be that the students had pipetted the positive control into the negative control well, or that something went wrong during the measurement. And using calibrated units, meaning having an expected value for a certain construct, actually allows you to filter for these kind of process failures. So a few examples here. Again, the positive controls had extremely high or extremely low fluorescence. The negative control was strongly fluorescence, or the negative control had negative values. So that is basically impossible. So that also means that maybe the students had understood something incorrectly, meaning it really allowed us to filter out these data sets. And process filtering is really different from kind of like filtering out outliers, like measurement outliers, because the process filtering really allows you to say, okay, something really went wrong in the process. We take out that measurement, and we measure the geometric standard deviation again. And that really helped in kind of like bringing down the standard deviation. So you can see that we now, if we filter out for the calibrated unit or the normalized units, we really go down to a reasonable standard deviation. So that means in principle, we compared 5 different ways. The last different ways relied on the fact that we had calibrated units and could really filter out these badly processed experiments. So that means for the first question, does unit calibration really matter for measurement precision, and how precise can we measure? It means yes, unit calibration, meaning using comparable unit, really matters. It allows to identify process failure. It allows to filtering out these process failures and to improve precision. And also, let's say in the context of synthetic biology, it could help for future data sets to indicate those process controls, to really tell other researchers, okay, this is kind of the margin that this plasmid or this device should perform in. If it doesn't, then something went wrong in your process. So we really think that it could be of value for reusers to also have these site process controls. Okay, so that brings me to the next study, where we actually compared cell count. As I mentioned before, we had now a way to calibrate fluorescence, but for the optical density, we would only use these spectrometer kind of calibrations. So how can we now actually come to cell count? Here we compared 3 different methods, and we also used again an IGEM Interlab to verify or kind of test which method would work better. In this Interlab, 244 laboratories participated. And as I said, they compared 3 cell count methods and applied to 8 strains that were constitutively expressing GFP in E. coli. The first methods, and just as the state of the art, so what are the existing methods for estimating cell counts? Of course, we all know it. It's optical density measurements at a wavelength of 600 nanometers. It's very fast, inexpensive, high throughput, and automation compatible. But the drawback is that it does not allow for good correlation to cell count. There is other ways to actually derive cell count, for example, microscopy, flow cytometry, or measuring colony forming units. The benefit is here that they can really distinguish or like measure cell count, often, especially colony forming units. They can also distinguish viability, so they can really tell you how many living cells you have in your sample. They're not affected by cell stage, such as inclusion body formation, protein expression, or filamentous growth. But they're, of course, not fast, inexpensive, or high throughput and automation compatible. So many of these methods actually take time. So we wanted to kind of like bring that together. How can we have something that is quick and inexpensive, but still gives us cell counts? The challenge of OD measurements or correlating OD measurements to cell count is actually the fact that OD measurements are based on light scattering rather than absorption. So you can see that here. So if a cell kind of like is hit by light, it doesn't absorb light, it actually just scatters light, and it scatters light away from the detector, meaning in a solution with cells, you basically have, it is almost a mimicry of absorbance. So, and the effect of the scattering is dependent upon the size of the detector and the distance between the detector and sample, which is kind of like just nicely illustrated in the sample. So depending on this light scattering, the number you get out from a cell suspension is really dependent on the specifics of the spectrometer that you're using. And also at high cell densities, you rather get these like multiple scatterings, meaning that that light basically is scattered back by a second cell into the detector, meaning that actually optical density measurements, especially at high cell density, do not correlate with cell count. So how do we now derive from OD 600 measurements in an easy, cheap, and high throughput compatible way to cell count? So the teams were first asked to do or to measure colony forming units. They were basically giving a protocol that outlined how they can use an overnight culture and dilute it several times, and then plate the last dilutions on plates such that you get enough colonies, but little colonies, or like enough colonies, but colonies that are actually countable. So that was the first method we wanted them to do, to derive from basically an OD to a cell count. The second method was using microspheres with E. coli refractive properties. So this method was based basically on a reported relationship between microsphere particle size count in OD. So microspheres are basically silica, monodispersed silica particles in water. And if they have a specific size, they are available in different sizes, and the manufacturer can also give specific specifications about them, about their refractive index. And if you choose the right ones, you might have silica particles that have a similar size to E. coli and actually have a similar refractive index, meaning they have the same kind of like light scattering properties as E. coli, but at the same time, you kind of can exactly know the number that you have per vel. So we basically gave them a known number of these microspheres that we chose, and then asked them to make a simple dilution series and measure that at 600 nanometers. And the last method was again this like colloidal silica that I mentioned before. It only provides a single reference value because actually the optical density measurement or the OD measurement that you get is so low that you cannot further dilute it. As I said, again, it calibrates for instrument differences. It's cheap and stable, but it cannot be used to estimate the number of cells. But we still wanted to see how it would compare against the other two methods. And then to determine the accuracy of the three methods, we basically calculated the ratio between the observed mean for each replicate set and the value predicted by the model fit. And as a model, we basically first used a model of perfect dilution because we did a simple dilution series. So in theory, you know very exactly how much molecules or microspheres or cells you should have in each vel. And for colony forming units, and actually I'm still having this in my way. Oh, sorry. And for colony forming units, we got, let's say, very bad results. So you see that here for the blue line. So basically the ratio between predicted and observed were very large, and basically we didn't get reliable measurements. So here we only compare colony forming unit, microspheres, and fluorescence because we also had them measure fluorescence. We didn't include the LUDOX in this model because we only had a single measurement for it. So for the microspheres, it already looked much better. So we got more precise values, as you can see here with the red line. But something that was, and here we just, as I said, used a simple pipetting model. But if we then in this dilution model included a pipetting error, so this is just a little term you can include in the model, in the dilution model, a term called beta that you can derive from by optimizing the fit. The fit to the model became even better. So this is why we now suggest to not use a simple dilution model, but rather a systemic pipetting error model, which is given here below, to fit the data of the dilution series to use it to calibrate your data. So this is what we came up with in this model. And yeah, with this systematic pipetting error model, we really got, we improved the standard deviation by several fold, and overall it's now only one fold, which means that the data points are really within percentages of each other. So it's relatively precise. It also means, and then given that the microspheres actually performed very well in this method, we actually wanted to know how that would compare to the true cell count. So the true cell count you can actually get from flow cytometry data, especially from calibrated flow cytometry data. And what you can see here is that the so in green and in yellow, and then, sorry, I'm stuttering a bit because I have this bar in my way, so I cannot see half my slides. So the values compare very, so the values derived from the cell count calibrated with the microspheres correlated very well with the true cell count that was derived from flow cytometry data, while the values for the colony forming units really were an order of magnitude difference. So this is basically given as molecules of equivalent fluorescence per cell. So that means that the microspheres were really, possible to give us very reliable data. And also an additional thing we found is that unit calibration really gives additional scientifically relevant information, namely how many cells are in your well. So, and also how many chromophores, for example, molecules of a fluorescent protein are present per cell. So, and this is what is now plotted here on the y-axis, and I just turned it around for you. You can see it a bit better. So you really get an estimation of molecules of equivalent fluoresceins, because often it's valuable to know not only an arbitrary unit, but really knowing how many cells are in my well and how much chromophores are they approximately making. The take-home message from the cell count calibration protocol is really that cell count estimation via plating is very laborious, variable, and systematically underestimates cell numbers. So it's really not very accurate. And as I said before, we really compared that across many laboratories, over 200 laboratories. So we believe it really, for many laboratories, doesn't work well to get reliable numbers from that. OD unit calibration with LUDOX is a reliable way to OD unit calibration by correcting for differences in optical properties of the spectrometer, but it does not allow for estimating cell count. But at least it's better for getting comparable units. But again, it doesn't give this extra scientific information for estimating cell count. And cell count estimation via microspheres, given one chooses the one with similar properties to E. coli, allows for precise estimation of cell count across laboratories and is really comparable to flow cytometry, which means you can also do data fusions with flow cytometry data. Again, in the context of synthetic biology, it would be really great if more data that has been generated can be reused by others. So it is a way to data fuse microplate and flow cytometry data. This is just what I said. All right, so coming to the last part of the webinar, where I talk about calibrants for blue and red fluorescence. Again, this was verified by a mini interlab. About 7 laboratories have participated in here because it wasn't possible to organize an interlab study during the pandemic. So here we were really using looking for chemical dyes that have certain properties. So first, we wanted to have a good spectral match for biological measurement target. So the match does not need to be perfect, but it should be good enough for approximation. The calibrants should be readily soluble in PBS or water, because materials that require other solvents, such as DMSO, increase handling difficulty across the globe. They should be widely available at low cost. So, for example, the materials for fluoresceins and silica particles cost under $1 US per protocol execution. We wanted to match that. They should be stable for room temperature shipping, and it should also match flow cytometer calibrants again, so we could do these type of data fusions in the future. So for red fluorescence, we therefore chose sulforhodamine 101, which is available via Sigma. It is affordable with €3 per milligram. It dissolves in PBS, and it has good spectral match with fluorescent proteins. And we found that a calibration curve of stretches over several orders of magnitude can be derived from using 2 micromolar starting concentrations and then using 10 twofold dilutions in PBS. And using specific excitation emission wavelengths. I want to say that we also looked into other calibrants that we had previously under consideration, Nile Red and PE Texas. They have good spectral matches, but Nile Red was difficult to dissolve, and PE Texas Red was actually too expensive. For blue fluorescence, we ended up using Cascade Blue. It is widely available via Thermo Fisher. It is affordable, although at a bit higher cost. It dissolves in PBS, and it also shows good spectral matches with most blue fluorescent proteins. And again, the calibration curve we measured should start at 10 micromolar, and then one can use 10 twofold dilutions in water. We also had looked into other calibrants that were previously under consideration, Pacific Blue and Coumarine 30, but for the same reason as before, they weren't very practical because they wouldn't either dissolve easily or were too expensive. And then just to show a few data, so we basically used them to calibrate several plasmids that we had shipped to the participating labs. And it turned out that the calibrants were easy to pipette and that the dilution series were very replicable. And this can be seen by the coefficients of variation for calibrant replica sets. The pipetting error that we determined in our pipetting error model was very low. And also the quality and fit to the model was very good. So there was no systemic issue with these calibrants. You could imagine that, for example, they could precipitate or would be difficult to pipette, but this is not the issue. And then we also, of course, used it to calibrate expression patterns from various constructs. Here, I just show the data for RFP. So you see that we get reasonably low standard deviations for these data. So those calibrants work well. Last but not least, I briefly want to also introduce or just touch up on this other paper that uses authentic calibrants for fluorescence, because it goes beyond what we have been doing. So here we of course use chemical calibrants because they're commercially available and can be stored. But ideally, you want to of course use the actual fluorescent protein for calibration, because the extinction coefficient for these fluorescent proteins is of course different than for the calibrants. And to really get detailed scientific information about the number of molecules per cell, you should of course work with fluorescent protein. And this study has actually achieved of doing this, because it basically provides protocols for purification and quantification of fluorescent protein calibrants. So very easy ways to get enough of these fluorescent proteins, and programs to also do the analytics of the workflow. The paper then also shows an assay to actually use unpurified proteins as calibrants and still get very accurate values. It also examines how cells can interfere and quench fluorescence. And eventually all of this then allows for actual quantification of fluorescent protein count per cell. So for the cell count, they actually used our Microsphere protocol. So again, this hasn't been tested within the IGEM community. I just wanted to tell you that it's out there, and we think it's really useful and goes even beyond what we're doing with our chemical calibrants. Yeah, so as take-home measures from the webinar, unit calibration for cell count and fluorescence yields lab-to-lab comparable measurements and enable data fusions of plate reader and flow cytometry data. Unit calibration also enables process quality control and thus filtering of low-quality data set. And this QC could be included in data sets for the future, such that other users can always know if the plasmids they're currently using are performing within the expected range, or if something in the process or protocol is not working. Unit calibrations also provide additional scientifically relevant information. For example, how many cells are in your well and how many chromophores you have as an approximation in your cell. Of course, using real authentic calibrants is always better than chemical calibrants, but we believe that our chemical calibrants are actually an easy way to get also this additional information. And then for cell count, we recommend to use the microspheres. For green fluorescence, we recommend to use fluorescent. For red fluorescence, we recommend to use sulforhodamine 101. And for blue fluorescence, we recommend to use Cascade Blue. I also want to give you some perspectives. So ideally, we would like to develop actually a calibration kit. So we're currently looking for a provider that sells the calibrants that are actually just introduced as a kit. So we could ship that to synthetic biology labs and they could use it as, as a workflow in their protocols. We're also looking to extend calibration to other cell types. Currently, we're only doing E. coli. It would be great to also use calibrate other bacteria, even yeast. We're also looking into ways how to automate data processing and all the calibration protocols. And of course, it would be great then for future interlabs that we're studying that now that we generated these calibration protocols, to actually use these protocols to generate calibrated unit measurements for existing parts in the IGEM distribution. So IGEM also has a huge resource of actual plasmid, promoters, terminators, and so on. So it would be great if we could use harness the community to help us in calibrating all these parts, so they would be available for the DBTL cycle and for the community in synthetic biology. And with this, I really like to thank you for listening, and I'm looking forward to answering your questions.
Fantastic. Thank you, Sonia, for a wonderful and exciting webinar. If you have any questions, put them in the webinar chat or in the Q&A. Before we get into that, some questions are already posted. This IGEM, it sounds like an amazing group that is working together. From the map that you showed, I noticed that there were some gaps in terms of global spread. What would people do if they want to get included in IGEM?
Yeah, I mean, IGEM is of course, an open competition. So I think the map was just showing the Interlab, but if you look at the actual map of IGEM teams participating, I would say there's definitely regions that have less teams, but it is a global effort. And also the 2018 Interlab was already more spread. So definitely, if you want to be included, you need to start a team. So the best way is to get started with maybe a supervisor in your university that is involved in synthetic biology or that you feel could be interested. And from there, you could start building a team, signing up, and participating. Yeah. So there's no other barrier than that, I would say. Wonderful.
Thank you for highlighting that. Yeah, related to that, I was wondering, since the pipetting error was quite a big impact in some of the measurements, do those teams get any standardized training in how to pipette, or are we just assuming?
That's actually a great and interesting question. So the teams do not get currently training in pipetting. So there's the IGEM competition, and then the level in between is the supervisors of these IGEM teams. And it really depends on how well they get trained in pipetting. But I think it's actually a good point to anyone out there in research is actually there is a skill in pipetting, and there is actually webinars and tutorials out there how to pipette accurately. It's also worthwhile to actually calibrate your pipettes very frequently. So definitely we did not provide that information, but I think it's also good because it gives a realistic, approximation across, the pipetting schemes and calibration of pipettes across the globe. But definitely take it as an opportunity to calibrate your pipettes.
Definitely. Also, maybe that one of these essays could actually be used for you to assess your own pipetting skills, because if you get a high standard deviation out of that.
Yeah, definitely. I think that's actually a great suggestion, because we're currently putting resources together to, we call it the IGEM how-to. So to have, many of the teams are actually quite inexperienced and they need some help to get started. It's a great suggestion to also use the dilution series, then fit it to the model, calculating the pipetting error, and then trying to improve it.
Yeah, beautiful. So to get to some of the more sciencey questions, in any of these measurements, do you think dead or dying cells can somehow confound the value that you get of cell number?
Dying the cells with a dye?
Cells that are dead or in the process of dying.
Or dying, okay. Definitely. So I think the microspheres, they don't, they're not, they can't measure viability of cells. So definitely, if you're reaching late log phase, you will get, you will count including a dead cell. So this is, that is a fact. So this is maybe also the reason why the colony forming units underestimates the number in comparison to any just, microsphere calibration, just because we really only measure the cells that make it all the way to forming a colony. Yeah.
Because that will be a follow-up question, right? Because that difference was, to me, quite striking. And the wondering is does that in any way impact assays where you routinely do that, like, for instance, a cloning assay? But if that is indeed can be explained by just low viability of the cells, then that would be...
Yeah. So in cloning, I guess, indeed, it could be low viability of cells. The question is, of course, how much does cell viability matter for these type of circuits that we're trying to build in synthetic biology? So how much, given, the bigger context, we want to calibrate the units to use those data to feed into our models to build better circuits. So how much does cell viability actually matter for the circuit output? That's a great question. Yeah, in this context, microspheres, they can't distinguish between live and dead cells, and we need to, it's a fact we need to take into account. Yeah.
In your first essay, when we were using the GFP, one question is, how long does it take for GFP to actually be expressed? And then does that expression in any way link to the promoter?
Yeah, so I think how long does it take to be expressed? That really also it depends a bit on the promoter, but I think the real rate limiting step is maturation of the chromophore. So I think that is really the rate limiting step. I think it also needs oxygen depending on the fluorescent protein you're actually using. So the promoter definitely determines how much mRNAs are made, and also that usually correlates somewhat to how much protein is made. So definitely the maturation, like let's say this is what we call protein expression. So it all comes together. Eventually you want to have kind of like a functional protein in the end and functional chromophores. So our assay, of course, measures the number of, active fluorescent proteins. How many inactive or maybe inclusion bodies you have, our assay can also not determine. So it's really, cross comparison of data. But if you cross compare, let's say, the same fluorescent proteins expressed from the same promoter, you could assume that they all follow the same, folding and maturation dynamics. But yes, of course, we can distinguish. Therefore, you would need to do a Western blot, RNA-seq. Definitely, you could dive deeper into the molecular numbers.
That could certainly explain why you might get different measures if you go to a different strain of bacteria, right? Just the processing of GFP is different.
Yeah, definitely. Yeah. So and also I think we will likely never solve this issue in biology, that the context dependency that we say, but at least we could quantify with this method, like how in our case we could quantify how many active fluorescent GFP we have in the cell eventually across different strains. But of course, we can't quantify why things are not active or why it takes longer.
Yeah. Could you elaborate a little bit more if you want to take this into the realm of absolute quantification? Why it is important that you use the exact same fluorescent marker that you want to measure?
Yeah, I think that has something to do with the extinction coefficient, I would say. So really, let's see. I would say it's really the extinction coefficient that makes the difference between really getting molecule numbers between a chemical dye and a fluorescent protein. Otherwise, you get kind of like estimates. This is why we call it like molecules equivalent of fluorescent. But to really get protein numbers, you would need an authentic calibrant.
All right, thank you. And I have some questions here on different proteins to measure. Like, could you adapt these assays to measure, for instance, chromophores that are present in a cyanide bacteria, or antibiotic resistance genes?
That's a great question. So of course, these chemical calibrants that we're currently using are made for fluorescent proteins. So one could look into chemical chromophores that have similar spectral properties than algae, like plant chromophores, for example, and then use those. But I think each case would really need to be evaluated how well it works, but I could imagine there is, chromophores out there. I mean, that you could even, given that there is now this authentic calibrant protocol, you could also work with chromoproteins. So there's a lot of bacterial chromoproteins that could be expressed that work rather on absorbance level than on fluorescence level, given you hit an absorbance peak that doesn't overlap with cellular absorbance or scattering. For antibiotic resistance, we actually have been thinking about it in my own lab as we work with antifungals. So a way to calibrate it to calibrate the activity, for example, of a resistance gene is using, for example, antibiotics themselves and using minimal inhibitory concentrations. So I think you can use our protocols as an inspiration to think about how you can calibrate a certain assay to the activity of a chemical calibrant. But definitely the assay would need to be adjusted to make it work. But I think it's always great to have some type of chemical that is chemically synthesized and available from a company that has a certain purity, and somehow make it comparable to your biological readout. And maybe then there is not more scientific information about it, but at least you can do these type of like process controls, and at least you can maybe give the data to another researcher and he can use it because he knows what these value means in equivalence to this small molecule.
All right, thank you. The ability to purchase from a company is of course also a great way in standardizing the reagents that you get. A related question to that coming in is do we have to mind storage conditions of any of these chemicals? Is that a point of concern?
Yeah, so for some of them, we have measured stability, at least as powder, and we have measured stability of the actual water or PBS solution. For the calibrants we used, I believe we have some data of stability over a few months, but we don't have data on really long-term stability, and also not like neither of the ready-made solution nor the actual powder. So that would also be something to be evaluated. Also, I want to mention in terms of precision and in terms of what I mentioned in the end of a measurement kit. So of course, all these teams were provided with solutions that were ready-made or ready weighed in. So I think also a cause of error could, of course, be weighing in the powder. So we didn't do the interlab on like how well teams can actually weigh powder and get solutions, calibration solutions out of this. So this is another source of error, of course.
That is an interesting question in itself, right? All these things that are standard in a lab that you don't realize are actually cause for moderate errors that do build up.
Yeah, indeed.
All right. in the end, you mentioned that you have an interest in tying it back to what you're currently working on, yeast, expanding these standardization techniques to other cell models or even mammalian cell lines, insect cell lines. Could you talk a little bit more about that? How you plan on approaching that?
Yeah, so I have been thinking a bit about yeast, where we could actually use similar microspheres because they are readily available in different sizes. So the issue a bit with yeast is it changes its size during the cell cycle. So it depends a bit on the distribution of which type of yeast cells you have in your solution. It might still be that the cell cycle is not synchronized; it just averages out. We haven't really tried that or worked with that, but this is a bit the considerations we have been thinking about. One could also think that it is a bit like growth state dependent, so that it's really important to take samples at a certain optical density or growth state. But again, we haven't really worked with this, but hopefully we can develop something that is easy enough and can ignore some of the cell cycle dependent size differences. But definitively, given yeast are also like different yeast types would definitely likely be needed to be calibrated with different microspheres. And the same is actually true for bacteria. So currently we really have looked into E. coli, but not much further.
All right. Thank you. I've got maybe one final question here is you mentioned a little bit being impacted by the pandemic. I was wondering if you could remark a little bit more on how the pandemic has affected your research, how you navigated certain challenges, especially as it relates to this work that you discussed today.
Yeah. So I think in terms of IGEM, it was, of course, as IGEM relies on teams working together on a wet lab project, it has really disrupted the work of many teams. Accordingly, also our planned interlab studies, because we basically couldn't do those. So for my own work, I was lucky enough that here in the Netherlands, we were only in a very hard lockdown for 6 weeks. And after that, we could go into shift work. So then people could work less in the lab, but at least we could still do some science. It has also the pandemic. So this is why we could still do this like mini interlab, where we basically just used committee members that had a lab available, and we just did the experiments. So that worked out. The pandemic has also affected shipments. So actually, because as we rely on a very global network and we need to ship components to each other, this was really difficult during the pandemic, and it remained difficult actually also for IGEM. That is, it's a US-based foundation to ship to China, for example. So there's really some new barriers in the world, especially for these like very global efforts. There's more customs control, so the plasmids often got stuck in customs for several weeks, which then again builds back to stability. So if a compound is stuck in customs for several weeks, how does it affect the quality of the compound? So definitely the pandemic has affected these very global efforts through ways that were maybe less anticipated. So not necessarily because we couldn't go to the lab, but rather we couldn't spread the components. We couldn't, mostly because of this, because we really couldn't ship anything to anywhere.
Thank you. Thank you for sharing that. I think it's an important one now that we're getting out of the pandemic, that we do realize that we have these major impacts that will probably resonate for some time in the future, and how we deal with it. So thank you for commenting on that a bit further. Yeah. That brings us, I think, to the end of the webinar. Renata, do you want to make some closing remarks?
Yes, well, thank you so much for a very interesting talk, Sonja, and this lively discussion. I think we had plenty of time to address all of the questions, and I hope it was helpful for everyone. If you have more questions, please feel free to visit our website, where we have a Q&A section where you can still post questions after the webinar. And as I mentioned earlier, the webinar will be recorded and will be posted on that website later, which will take about a week or two until it's been uploaded. So check back in a little while. And lastly, I would like to announce our next webinar, Designing and Executing Prime Editing Experiments in Mammalian Cells by Jordan Doman in David Liu's lab on May 16th. If you want to know more about this webinar, please visit our website. And thank you again for everyone for coming and joining us at this webinar. And I hope to see you soon.